Hello, everyone. So let's start from the second lecture, lecture 2, talk about microscope and stain technology. This part, I will be go over relatively quick for the slides because a lot of them will be talked about in the lab. So go to look at the lab video also because it's very practical. Okay, so we'll go very quick on the slides. So microscope. We talk about a microorganism. We say it's too small to see. So we rely on microscope. Now, what are the microscopes? How to characterize them? We have light microscopes and the electron microscope. Now, light microscope including bright field, dark field, phase contrast, fluorescence, and the laser confocal microscope. Electronic microscope including AEM, which is transmission electron microscope, and the SEM, scanning electron microscope. Now, we are in the lab, we are using actually is bright field microscope. Now, why we want to use microscope? Because we want to see the bacteria clearly. So here is a concept called the microscope resolution. So that is the ability to differentiate the small objectives that they are very closely attached. So you see the picture here. This is the points are unresolved. This is partially resolved, and the last one is fully resolved. Now, how we do that? We will talk about in the lab a detail, which is calculated by lambda divided by 2 and NA, or P equals lambda divided by 2 NA. So based on that equation, we know the wavelength, if it's shorter wavelength of the light, will generate greater resolution. And you can see here the resolution in light microscope compared to the electron microscope. This is electron microscope, here is light microscope. You can see the difference between that. It's more clear, more differentiate in the electron microscope compared to the light microscope. So first is a bright field microscope. We will talk about the structure very detailed in our lab. Okay, basically including ocular lens, objective lens, stage, coarse focus, fine focus, iris diagram, condenser, light resource. Then what is the bright field microscope? Is dark image, brighter background. And we talk about is a parfocus. That's a very important characteristic for bright field microscope, which means once we focused, the microscope will be stay focused. We only need to do a very minimum adjustment using fine focus. We talk about the total magnification, which is multiple results from the ocular lens, the magnification multiplied by the objective lens. Now we talk about oil immersion. Why we need oil immersion? We put one large drop of the oil on the slides. Because the light passing through the resource or from the glass, go to the air, go to the glass again, it will be reflected. And the refraction will causing this clear of the images. So we need to add one drop of the oil to remove the air gap. Therefore, the glass oil will be all together because the refraction index of the oil and the glass are the same. Now, dark field microscope. Dark field microscope is actually, we had a block underneath the condenser. So the light passing through the specimen is perpendicular. Can you see that? It's perpendicular. They can see, kind of we will find is that the background is dark and the specimen is showed up. That is dark field microscope. Now, dark field microscope is using for testing a group of special bacteria called spiral sheets. This is one of the example is Treponema palladium. T is special name, palladium is general name. This is a pathogen called syphilis. So you can see the dark field microscope background is dark. This specimen showed up. It is which is twisted. That's we call it asexual filaments. It's called spiral sheets. Okay, phase contrast microscope. Phase contrast microscope. The tricky thing here is that phase ring. Because of the phase ring, so the light passing through the specimen will be either 1/4 front or 1/4 behind. So they will be showed up because the light peak at the bottom from the spectrum, it will be difference compared to you do not have a phase ring. Okay, now phase contrast microscope is using to see the internal structure of a microorganism, most of the time for eukaryotic cells. So for example, Saccharomyces cerevisiae, this is a yeast used for beer brewing. So we have the quite beer yeast. And you can see that this is a Bright-Field Microscope. You didn't see anything. Right side is Darkfield Microscope. You can see a little bit of internal structure, but in the middle is Phase Contrast Microscope. You can see very clearly the structure in the middle. Okay, Fluorescence Microscope. Fluorescence Microscope, in difference from light, in difference from what we are talking before is all those light field microscope. We talk about the bright field, dark field, phase contrast, their energy resource is light. But the fluorescence microscope is a little bit different. Their energy resource is UV light, violet, and blue light. Now the second thing is that the specimen, they must be stained. Then the stain is not like those general dye. They are using fluorochromes to do the stain. And more important here is this mirror. This diachromatic mirror will be reflect short wavelengths and transmit longer wavelengths. And together with this barrier filter, only longer wavelengths will be passing. Okay, so they are applied very specifically in medical microbiology and ecology studies. The good thing is using fluorescence microscope, we can differentiate a dead and alive cells. Now, what are the dye we usually using for the fluorescence microscope? And we mentioned we have to label them first, use fluorochrome labeled products, including antibodies and the fluorochrome dyes. Here are some of the fluorochrome dyes. We use a lot is the DAPI, D-A-P-I. It's a DAPI stain. We call it DAPI stain. That stains the DNA. Okay, so because of that, we can stain the DNA, so we can differentiate live and dead cells. Most of the time, the dead cells is red and the live cells is green. So that's an advantage for that. This is some picture you can see the fluorescence microscope. Okay, this is what we see here is actually, you will see it's bacillus. This is streptococcus. Streptococcus is like a chain shape. And this is a yeast. We call it a valve. Okay, confocal microscope. Confocal microscope is we are using the laser. The laser usually we call it argon laser. It's coming from a company called VES from Germany. The good thing for the confocal microscope is that they using laser beam. So, and connected to the computer system. So they will generate a 3D images. All the others are only 2D images. Confocal microscopes that can generate 3D images. And very good to using to see the biofilms. So what is the biofilms? that is a matrix of the bacteria accumulated on the surface. Because they accumulate each other, they transfer their signals, we call it chlorine sensing, and they will be stacked together and very strong and resistant to the sanitizers. And that's a risk for the meat and food industry, especially for the food contact surfaces. So that's where we need to do something for the biofilms, and the confocal scanning laser microscope is a good tool to do the observation. So this is a confocal scanning laser microscope, what I did when I was a student for the research. So the first part is that you see, it's also need to stain. Okay, so we use, we call it a Cyto 9, a dye kit to do the stain. This is a control. We see lots of the live cells, so it's a red color. Then the color spectrum, which is on the right side. So x-axis. The second way, which is we clean the surface using the distilled water. And you see some of the dead cells there. Can you see there? Some of the red color, that means dead cell. Then the color spectrum, corresponding to that, we see the y-axis start to show up, which means the color start to become red. This is the one we use, the third picture we using the chlorine. So we see more red cells because most of them are dead. And the last one we using chlorine with surfactants. You will see only 2 colonies still survive. Most of them are brownish color, which means it's injured. And you can see the color spectrum, which is right in the middle. So that's used laser confocal microscope can see live and dead cells for biofilm on the food contact surfaces. Okay, smear preparation. This is what we're going to practice in our second lab. Actually, in the first lab, we will do the simple stain. So how to do that? We will be making a very thin specimen on the glass slides. It doesn't have to be thick, because if it's very thick, we will not see the morphology. Second, we are allowed to have a completely air dry. If we don't do the air dry, we will see a lot of legs on the images. The third step is we call it heat fixing, because we want to inactivate enzyme, let them stack on the surface. The last step is we do the dye, which is adding a 1 drop of the dye in the middle. Then we do the observation underneath microscope. That's called the simple stain and smear preparation. Now, what kind of the dye we are going to be using for the simple stain? Why we can do simple stain? Now, first of all, you need to know if the bacteria, they are growing overnight, they usually it's negative charged. And then all the basic dye, which is positively charged. So they can combine together, attached together. This is called a simple stain. So the simple stain we are using is crystal violet, saffron, methylene blue, and the carbon fusion. That's all the dye we are using. Now we have to be careful about methylene blue, because methylene blue does not have a very good attachment. So unless you have a very good experiences, we don't use methylene blue too often. Now, sometimes for some bacteria, we also using acidic dye, which is negatively charged. So the dye is repelled for the bacteria. Instead of their staining the bacteria, they stain in the background. So that's called a negative stain. A negative stain including nigrosin Indian ink, earthin rose bagel, and acidic fusion. Nigrosin and Indian ink are the most 2 common dye we used. So this is the simple stain. After simple stain, we could see this is a crystal violet stain of E. coli. And this is methylene blue stain of corynebacteria. Corynebacteria is stacked together, we call it club shape. Because simple stain, we know the size, shape, and the arrangement of the bacteria. That's why we have to do a simple stain. This is a negative stain of the bacterial capsule. And this is actually Streptococcus ammonium, which is a lancet shape, is a short chain, and the capsule is a polysaccharide, very heavy packaged. So we see the background is dark, then the specimen showed up. Now we're going to talk about the differentiated stain, which is gram stain, acid-fast stain, and some special stain, including endospore stain, flagella, and the capsule stain. Now, gram stain and endospore stain we will practice in the lab. The others we'll just talk about that. Now this is gram stain. So we'll talk about detail, the practice. 4 steps: crystal violet, followed by gram iodine. To make crystal violet stain more strong, then with the discoloration, we use alcohol, and finally counterstain with the safranin. So gram-positive bacteria stays purple or blue, and the gram-negative bacteria stains pink or red. And this is a way to differentiate all the bacteria to become gram-positive and the gram-negative. Now for the yeast, I want to mention, we do not say gram-positive or negative, we all say gram-stain blue. And it's a differentiated bacteria. It's a differentiated stain. And also we mentioned in the lab, as a gram stain, we need to record 3 important information, which is gram results, positive or negative. Second one is morphology, it's a cocktail or lods. Last one is arrangements. Is that a single or a chain or grape shape? That's 3 important information for the gram stain. Now, acid-fast stain. Acid-fast stain is using for testing a very special bacteria called microbacteria. Because microbacteria have a mycolic acids, which are the short chain unsaturated fatty acids in a cell wall. Because of the mycolic acid, so the cell wall is very waxy, so we call it a waxy cell wall. So the general dye cannot come in. We have to use carbon fusion with heat steaming to let the dye go inside. And this is used for testing microbacteria tuberculosis. We know tuberculosis causes a lung disease, a severe lung disease, okay? And the microbacterial lepro will cause leprosy, which is actually the bone fraction. Now, endospore stain is to stay the endospore. Now, there is a 2 types of the endospore stain, which is a negative stain we just mentioned, and also directly stain. Capsule stain is to use stain the bacterial capsule. Okay, it did not show up here to stain the bacterial capsule because capsule is usually composed by polysaccharides, which is very important for the pathogenic attachments. Now, flagella stain, we are using a very special treatment to stain the flagella, which can be showed up because flagella indicating a real movement of a bacteria. Now, all these 3, we will talk detail in the lab section. So go ahead to look at the lab video. Now, this is something showed up here is gram stain results, acid-fast stain. We see the blue is non-acid fast, red cell is acid fast. Endospore stain, okay, endospore stain for bacillus. Endospore stain specifically for 2 types of the bacteria, bacillus and the clostridium. This is capsule stain. And the last one is flagella stain. This is the Proteus vulgarius. We're going to use a lot in the lab. You see the flagella covered on the surface, which is we call it a petrotrichus, which means they can move around dramatically and have a very strong motility ability. Now, electron microscope, we have transmission and scanning electron microscope. We will go it easily. There is an electron beam right there. There is 2 different methods. Transmission electron microscope. The magnification can be as much as 50,000. We see clearly inside the structure. Scanning electron microscope, we only see the surface because the scanner is back and forth. So the magnification is about 7,000. We can see also the 3D images. Now, electron microscope, we will not do anything in the lab. Usually it's a shared facility at WVU has the electron microscope, require you have a special certification. So we won't go too much deeper right here. This is the transmission electron microscope. You will see the cell wall, ribosome, and the nuclear gases. This is 3D images for some small objectives. Okay. Now, why we need to do the stain? Because we want to see the shape and the arrangement. So what are the shape? I give you example is cocktail. This is Streptococcus agalactiae. We call it a honeymoon bacteria called sexually transmitted disease. So that's a cocktail in a chain. This is Staphylococcus aureus. We did in the lab. It's a cocktail. In cluster, looks like a grape shape. So based on the cocktail, we can say diplococcale, it's pairs. Streptococcus, it's a chain. Staphylococcus is grape shape. If 4 of them stack together, we call it tetrads, like a Micrococcalidius. And if 8 of them configuration together, we call it a Sarcini. This mole right here is bacilla, which is a large shape. For example, we also call it Coccobacillus sometimes, which means very short rods. When we move on to the examination 2, we will talk about the special bacterial pathogen. Lots of them are Coccobacillus. Let's say Haemophilus influenza. And the vibros, which is a comma shaped, let's say Vibrochloreal. This is the one will cause lots of damages for the seafoods in the ocean marine area. Clostridium, this is Campylobacter jejuni. This is sparrow-shaped spirium. This is a pathogen together with Salmonella, the high risk for the chicken products and the poultry industry. Spirochetes, we just mentioned, the special bacteria has asexual filaments. Leptospira interrogans, which is hook shape, will cause jaundice of the eye, which is yellow eyes. That's a spirochete. And this is underneath Darkfield Microscope. This is Bacillus bacterium, which is large shape in the chain. Now you can see compared to the E. coli, both of them are large, but the Bacillus are large. Okay, so we end of this section very briefly. So more detail we'll talk about in the lab.